we'll kind of.
Sorry, Tara, I'm just going to start recording.
Good. I think we've got a good number of participants here. So just to introduce our special guests, we have Dr. Enrico Bonello from Ohio State University. He's a plant pathologist, and Connie Hausman, a senior researcher from Cleveland Metroparks. And we are thrilled and so thankful that you could be here. Early on in BLD, myself and a lot of colleagues that I spoke with kind of felt like we were in the dark with this monster and didn't know how to proceed. And thankfully, we've been able to get more information. But really, the key thing that we're missing now is what the trajectory is going to be for the next several years. And since Ohio experienced it several years before us, we're hopeful that you can kind of enlighten us.
I know Maria Muscoli in the group because she's part of our BLD coordinating group, research group. Hi, Maria.
Well, thanks for having us. I know that this was put together, and I do have slides. So I actually just came back a few weeks ago from giving a BLD, basically everything in the kitchen sink kind of a talk at Penn State Extension to their Winter Force Health meeting. And so that's a huge presentation. Not that I plan on giving everything there, but for my benefit, I guess part of what I think might be good for both Enrico and I to hear is just sort of nod a head or thumbs up just to confirm that everybody is aware of beech leaf disease, has seen it, has some experience, et cetera. Okay, because that'll be a lot easier to not have to be too repetitive. And so, Enrico, if it's okay with you, I mean, we could just pause and ask if there's any pressing question immediately that you would like us to talk about first. Otherwise, I'd be happy to just share a little bit about our experience and monitoring and survey efforts.
And just to I suppose, clarify, my expertise really sort of on the biology and microbial interactions side, and also in devising potentially new detection methods. The field experience and the management really is with Connie. So maybe bear that in mind as you ask your questions.
So one question, Dr. Bonello, because I know that you had done research that indicated that perhaps that the nematode is more a vector than the cause, that there are bacteria and was it fungi too that are present in infected trees that are absent in uninfected trees? Is that the case?
So we found one fungal species that was unique to the symptomatic plants, but we did find 4 bacterial species that were associated with symptomatic plants. So that's in association with the nematode. So the nematode seems to be a necessary but perhaps not sufficient condition to cause a disease. But we don't really know because nobody's really has been able to do any experiments where they have inoculated plants with clean nematodes that did not carry anything. So we don't know, we really don't know at this point. Again, as I said, the nematode seems to be a necessary but not sufficient condition because you find the nematode also in non-symptomatic plants and in areas where the disease is absolutely unknown in some cases. The loads can be different. So in general, you see much more nematode in the symptomatic plants than the asymptomatic plants. But because you have this sort of overall distribution of the nematode that is also in non-symptomatic plants, the linkage to being a causal agent is still a little unclear, right? Because of that. That's the only thing. That's all we can say at this point.
I do have a question.
Yeah.
I was reading an article that my mentor sent me, and it was talking about how like some symptoms I mean, the nematode may be present like before the symptoms. So what you were saying, like, yeah, like, is it something in the nematode or is it like the nematode itself? I don't know how to like put the words to
Yeah, when
explain.
I say causal agent, is what is it actually causing the disease,
Mhm.
right? Is it just the nematode or is it the nematode plus something else?
Yeah.
That we don't have the answer to that at this point. But it seems like you cannot have the disease without the nematode.
Mm. So it's like something that infects the nematode that like infects.
Potentially, yes.
Oh, okay.
Or not necessarily infected. Well, the word infection has different connotations. Something that the nematode carries with it, either internally or on the outside of its body. Some microbe that contributes to the disease. It might well be what we call a species complex, where you need all of those things together to cause the disease. We just don't have the exact answer at this point.
Dr. Bonello, one of our participants, Bud Vaverka from Mianus River Gorge, and he's here, so if he wants to describe it, that would be great. But he was talking about a property in Pennsylvania where BLD has been present for years and hasn't progressed, which is quite different than what we're experiencing here in Lower Westchester. I don't know, Bud, do you want to describe that briefly? And then Dr. Bonello, if you have any thoughts on what might cause different severity levels, that would be great.
Yeah, it's an upland forested area, probably about 25% beech. And the site itself is right across the line. It's in Crawford County, Pennsylvania, from Ohio. And every year we see the signs of the BLD. We see some of the leaves withering. Even some of the branches will lose the leaves during the summer, particularly by August, and then the next year it's all back. The leaves are all back. We haven't seen over 6 or 7 years any real dieback. And I don't know. And then I go to other areas and you do see the young trees dying. So I don't know what the factor is, but it's not like it's not in a wetland or anything like that. It's fairly open, rolling hills, Western Pennsylvania kind of, landscape, just kind of upland. I don't know. And it made me have questions about what is the mortality. We're, seeing it 7 or 8 years, you would think you'd start to see something and some of the trees starting to die, but they don't appear to be that way.
So I will let Connie answer that question because she's much more better positioned, but I want to ask you though, Taro sort of introduced you by saying that you don't see the disease expanding. So those are two different things. Is the disease not expanding and or are the trees just recovering, right? Those are two different things. The same trees, in other words. I mean, the disease is not expanding for some reason and the trees keep recovering and they're not dying.
Yeah, I'm just seeing the trees recovering. Expansion, it's a small property we have in up there. It's my family's property. And I walk it every year. I'm there in the summer, the winter, and then I'm, back the following summer. And none of the trees, and I, as a biologist, I walk through the woods and I take a look and I've been seeing it for years. And I'm like, and that's how I knew it when it was showed up here. I knew what it was already.
Yeah.
But
so the question really is about
I'm not seeing the dieback that is described in some of the papers.
Yeah, I'll let Connie.
Yeah, so there are stand-to-stand variabilities. And so most of the dieback that we're still seeing, and I can jump in and share some slides, and our monitoring project is still in smaller trees and young saplings. So it depends on that structural diversity within your stand. But even in the areas where we've had BLD for 8 years or more, we are still finding trees that look fairly good. Nothing remains totally asymptomatic. So if that's a possibility in your brain that your fingers are crossed, then I apologize. That's not true. Everything gets some degree of symptoms, but they don't all decline at the same rate. And you can see year-to-year differences as well. And those are maybe environmental stressors additionally, but we are seeing some stand stand variability, and those potential tolerant or resistant trees, we are mapping and trying to monitor and collect scions from to be able to do some, experimental projects on, and that's in partnership with the U.S. Forest Service and Holden Forest and Gardens. So that's awesome that you potentially have a site or a stand that would be good to monitor and track through time. So I think I'm sharing a screen.
Mhm.
What do you guys see?
We see it.
My presentation?
Yep.
Okay, I'm split screen, so sometimes I get a little bit wonky moving my mouse around. So as I had mentioned, I just came back from a presentation in State College, so I'm going to throw a couple of slides. I'm happy to go super fast. I'm happy to pause. I'm happy to try to rapid fire scoot through.
And Connie, this is Brent, just to interrupt, because you were just on a topic that I know our group is interested in. If during your talk, if you wanted to talk about the process through which you're looking for what we would call like the lingering beech or the surviving beech in an area, we have a similar program here in Lower Hudson, and it's actually statewide as well, for lingering ash. And I'm just curious whether you have a protocol established for that, and we would like to hear about that either during this presentation or at some point during this call.
Yeah, no, that's great. And it's Jennifer Cook with U.S. Forest Service. So
Okay.
Same thing with the lingering ash. She's very interested. It's she who's also partnering with Holden Forest and Gardens, setting the threshold for what a particular tree resistance looks like. I mean, some part of it is how long has BLD been in that area? And then what is the general condition of that tree? And so we picked stands when we were looking for resistance that had been exposed, at least 8 years, because we really wanted to see mortality in saplings, know that it had been around, and it was clear that whatever the nematode population load was well in the environment, well experienced by as many trees as possible. But that's in development. That's all still in development as well. So
Okay.
This is probably.
How are you
Go ahead.
establishing the nematode load?
No idea. Lots.
You don't know? Okay.
Thousands. That's. Go ahead.
Yeah, I can help with that if you want me, Connie.
Yeah, go right ahead.
So we, well, first of all, because the crowd here may not be entirely in tune with the concepts of resistance, I just want to mention that resistance does not necessarily mean that the tree is completely free from disease, because resistance is what we call a spectrum, or it's sort of a range of responses to the pathogen. So really the most important thing for us, I think, and Connie, I'll ask you, from an ecological perspective, is that the trees are surviving and reproducing. If they have some symptoms on them, that's okay, right? They're still resistant. So they don't have to be completely clean of disease, although that would be the preferred phenotype, okay? But if you're looking for resistance, make sure you don't confine yourself to just the super clean, so only the asymptomatic trees. As far as the nematode load, the way you do it, in the lab, you would do, you would put leaves essentially underwater overnight, and the nematodes swim out, and then you can count them, sort of thing, okay? But we also have developed a new PCR-based tool or method that can be used to quantify the number of nematodes in the leaves. And if you're interested, I can show you some stuff about that later, but that's how you do it.
Great. I really appreciate you tackling those questions. We can talk more about it later. I didn't mean to interrupt you, Connie, but you were on the topic, so I just wanted to.
Well, and this is, and like I said, I'm so happy to keep it sort of free form, and then I don't necessarily have to go through slide by slide. I can show you a slide sorter if there's something in particular you guys want to see. But
Yeah.
Echoing Enrico's sentiments, I mean, it's the same thing with the lingering ash. It doesn't necessarily mean that EAB isn't going to lay eggs or larvae aren't going to get inside that tree. But they end up having the capability of dealing with that. So I'm just going to show, this is the national BLD range map. Cleveland Metroparks has funding from the U.S. Forest Service to produce these maps. So we collect all of the detection records across the state. There's a couple of different ways that those records are collected state by state. But this shows you sort of the chronology through time. You can see the yellow county in Northeast Ohio, that's Lake County. Just to the west of it, this is Cuyahoga County. This is where Cleveland Metroparks is. And so, early on, we were, experiencing symptoms before many others were. And so this just sort of is a nice illustration of, one, where I am. This is where I was in Pennsylvania, but just illustrating, these detection records and locations as far as our current knowledge. And so, as of 21, there's, 49 new counties. And it basically just represents this exponential growth of detection. Now, whether or not that is actually spread, or just because many more partners like everyone here is looking for it and reporting it officially. Because no, I seriously doubt that any of the BLD that was in Northeast Ohio spread to Long Island and caused that effect in 2019. Based on the records in Long Island and Connecticut, when they were showing us images of their BLD, they likely had it for at least 3 to 4 years before they reported it in 2019. So that's the current status. And so I'm sure that's all why you're here and what we want to know. I like this particular map because it actually shows where people have looked and didn't find it. So this is also the importance of those early survey records. And like I said, sometimes it's done through state agencies, and Maria contributes, and they do so through their own app. And sometimes we have a free app called Tree Health Survey that also helps collect records for us. And this is the app. It's like I said, it's a free downloadable app that Kent State University developed, and we've been modifying. We need some new grant money to be able to make sure we tweak some of the user access. But this is a platform that you all, since you're not part of the state and tapping into their Survey 123 related records, you could download this app, submit those records. And I think it was maybe Bud, to your point, one of the things we also do with this map is we actually will map severity. And so I do two things. If you look into Northeast Ohio, there's dead trees. So we record the location of dead trees. But in a year-to-year basis for this resistance question, we actually have people out on the landscape in this heavy hit long-term BLD sites looking for those trees that look pretty good. So they do a condition assessment within that Tree Health Survey app to help us understand where those locations are. So in Crawford, County, those records could come in as well, as far as saying, these trees are in pretty good shape, and we'd be able to track that through time. Yeah, so this is just a citizen science-based kind of model. So I'll pause for a second because I can jump into some of our monitoring if you want to see how we've been tracking it through time. Yeah. Okay. So Cleveland Metroparks, for those of you not aware, our metropolitan park systems are actually political subdivisions of the state of Ohio. And even though we're Cleveland Metroparks, we are the Cuyahoga County's park system. And we actually have property in 6 counties in Northeast Ohio. We were established in 1917. So it's a little bit of a misnomer if it's just Cleveland. We're not a municipal park system, because we own and manage over 24,000 acres. And as part of that effort, we have a long-term vegetation monitoring project in which scattered across 400 monitoring plots. And we sample these at 100 plots a year for 4 years. And that was started in 2010. And then we repeat on a 5-year cycle. And so I had data across our park system, because these plots are spatially balanced for all of our beech population. And like I said, BLD was first detected in 2014. So within this data set, I have almost 3,000 trees and 2,600 shrubs of beech that then I can go back to look at what was their condition before and what's it look like now. So here's sort of the status of our monitoring. And again, these are, you know, the 100 plots a year for these 4 years, 2015, 16, 17, and 18. And you can see the proportion of BLD by plot. So of 100 plots, right? And so after, you know, 6 years, we've got 85% of our plots have BLD present. And in 2021, we actually repeat sampled the 2015 plots. Now recognize we only had BLD in our parks for 1 year. Because we first detected it in 2014. So this is only 1 year with which we've had BLD. And when we came back to resample it, 87% of those had BLD. Now, that's just by plot. So then if we look at that by stem, 87% of the stems were affected with beech leaf disease. And so it's scattered across, it's spread across our park system. It's affecting almost all of our plots and nearly every stem. So that's the Debbie Downer side. And this is kind of what it looks like. We take photographic monumental pictures. So this is 2011, before BLD was in our park system. And this is the same plot 5 years later. And it's right about the same location on that slope. So this is what it looks like. Now, from the mortality perspective, I know this was a big question of our small saplings, because everything is still fairly small at where we're seeing mortality. It was 7% in 2018. We were at 10% mortality of our small saplings in 2021. But we're seeing an increase in size of our DBH size of our trees that are experiencing mortality. And they're increasing in frequency as well. So, and those are just on our monitoring plots. And that's what's also challenging, is because, you know, large trees are also dying. I mean, I've got really large BLD trees that are, you know, over 20, 25 inches DBH that are also dead, but they're not in a monitoring plot. So, you know, it's a little hard to say, and whether or not there's other, you know, stress factors on the landscape sort of tipping the balance.
Connie, what did you mean by on the stem? If you go back.
You want me to go back?
Go back again. Yep.
Here?
Yeah, 87% of stems affected with BLD. What do you mean by that?
Of the beech stems.
Yeah, but like, what are you looking for there?
Any degree of BLD symptoms, right? So it can be just mild leaf symptoms present, or it's that interveinal banding pattern of the leaves that we look for that are our typical BLD symptoms.
Okay, so yeah, that's
I
what I
wonder
mean.
if Brent, yeah, I wonder if Brent is not getting mixed up between stems and trees, because that's the same thing.
Mm-hmm.
So it's incidence.
Incidents,
Yeah.
right.
Okay.
Trees.
I got it. Okay, I thought you were talking about there. You were looking at the stems somehow to get the diagnosis as opposed to the leaves. I just wanted to clarify that. Okay, gotcha.
Right. Pardon
Okay.
me. Yep. So let's see what else. Oh, you know, I'll pause for a second, because like I said, I can keep going. I can sort of exit my slideshow. This is in 2015, just as a nugget. It didn't seem like this was going to be a very good thing on our landscape. And so I selected a subset of our plots that we would monitor annually. Because again, most of our plots were set up to be cycled on a 5-year basis. And I did my PhD work on emerald ash borer affected stands. And so I knew that there were going to be things that we were going to want to track, and it can move pretty quickly. But so we looked at across our park system, and again, this is the outline here, reservation by reservation, which ones were affected in which year. And I picked a subset of plots that we were going to follow and track on an annual basis. And so it gave me about 300 total trees that in total, and it was 13 plots. Some of them had BLD a little bit, and some of them were asymptomatic. So I wanted a range of original expression. And I wanted to be able to see all of the trees. So that was also a limiting factor. I had to make sure that each plot didn't have very many trees so I could see full canopies to do proper canopy assessments. And basically, and I know my tree size divide here is 5 inches. I know a large tree is not 5 inches, but it's a really nice break in basically what's a small sapling. So we divide saplings into things that are less than 5 inches and large trees that are over 5 inches in TVH. And through this annual monitoring project then that we did, you can see the decline. The overall average canopy cover declines much faster in those small trees. So within a few short years. And I have the x-axis is years with BLD because I had earlier vegetation monitoring data. I knew exactly which year those plots experienced BLD for the first time. So that's the years with BLD exposure, because that really sets the time sequence for how quickly the trees decline. So again, I will pause. I want to be respectful of everybody's time and questions and see if there are any.
Yeah, I have a couple questions.
Sure.
So how much time can elapse before the banding sign is showing? Because that's usually how we detect it. And I just wonder, how many years could that tree have been infected before showing that signs, or is it the same year?
Oh gosh, that's a giant question mark. So Enrico's shaking his head because he knows that nematode DNA is detected in trees that aren't showing signs of BLD symptoms. So we don't necessarily know how long that tree could have had nematodes in it before it has that expression.
But the cause of mortality would be from repeated leaf drop and photosynthetic deficit, I'm guessing.
Yeah, so let me fast forward here. I'll just do all of these real quick because essentially what we're seeing, if we break up our canopy cover into these 3 different conditions, either the leaves are asymptomatic, and that's the far left. They have some mild form of banding pattern, which is usually less than 2/3 of the leaf surface covered. And then the heavy BLD symptoms, the thickened, darkened, leathery texture, crinkled kind of leaf. And on a year-to-year basis, asymptomatic cover declines, but the banding and the crinkling can be highly variable. And that might be attributed to how much nematodes are in each bud on a year-to-year basis. And with a project with Enrico's student Carrie, we showed that, the leaf condition and bud in 1 year doesn't necessarily predict what the leaf condition is going to be the next year. And so it kind of depends on how readily the buds are re-inoculated by the nematodes within the environment. But the last graph here is this fine dieback, and what you'll see in this picture that I took below, this is a really large beech tree that doesn't have large main dead branches, but a lot of thinning. It's the fine dead branches, and that's that repeat stress and the aborted buds that you have year to year to year that basically continues to decline that tree. So it's not dead yet. But that's sort of the threshold. Is it, capable of recovering, or is it in a fatal decline?
Do you also have beech bark disease out there?
So we do, not as heavy as I think some of you all have had beech bark disease. And actually, I'm going to end this slideshow because I know Maria is on, so that I can jump ahead to this particular project. So you all in New York had partnered with us in some long-term monitoring plots. And this is that standardized protocol that Maria does across the state. And we did a joint paper with Sharon Reed in Ontario that basically was assessing the beech population in these plots for beech leaf disease, but as well beech bark disease, and whether it's the scale or the fungus or both, trying to see if there's an accumulation of factors that then sort of tip the balance. And in areas where you have beech bark disease previously, does it mean you're going to be more susceptible, or is it going to decline faster, et cetera. And so beech bark disease for us in the Cleveland area, Holden Arboretum in Geauga got it real bad, and they had a significant amount of mortality. It did reach our park system. We had some limited effect, but we still have I said have. We had a really healthy, robust, mature beech population. But for us right now, the beech leaf disease is pretty bad. So this paper that was published with this monitoring data, it is much more focused on the Great Lakes region, because we know that this pattern may not necessarily be the same when it comes to comparing beech bark disease plots in the mid-Atlantic states. And so this was, like I said, part of our long-term monitoring project. There were 60 plots set up across New York, Pennsylvania, Ohio, and Ontario. And I can just fast forward a little bit. These were the main takeaways from this particular project, because we did look at the percent of trees with BLD, and then with scale and fungus between the United States on the left and Ontario on the right. And really Ontario ended up having beech leaf disease was way more common for us in the United States than it was for them. And likewise, they ended up having, and it affects all size classes. So that's typical. But the neonectria was fairly uncommon in both areas. And so there's differences here, and not to say that necessarily one affects the others per se, but it is something that we're trying to, tease apart right now.
I have a question.
Mm-hmm.
Beech leaf disease can be fatal, right? Like it can kill the tree.
We're documenting mortality in small saplings for sure, due to
Oh.
beech leaf disease alone. Yes.
Okay, so is it like when it gets severe, is it something that like spreads to the rest of the tree? Like it spreads all over the tree, I guess? Or is it just stay in the leaves and that's how it like declines?
So what you see is the banding pattern continues to get worse and worse in the tree, and you have dieback. So fine twig dieback, basically on a year-to-year basis, it continually stresses the tree. And Enrico, I don't know if I know of any projects that are really honing in on looking, at decline or loss in photosynthetic capability. But I know that the mesophyll layer in these leaves is hugely damaged. And so I got to imagine that you're sort of stressing the tree out there. Do you want to chime in?
Yeah.
I would say that it seems to me that the damage really comes from the eventual killing of the buds, right? So that there's no new foliage being produced. It's not a typical dieback where you have the tissue being killed by something, like a fungus producing a dieback. It's the buds that actually get aborted over time. Is that right, Connie?
Yeah, right. And so you have fine twig dieback and fine twig dieback, and then branch thinning, and then it just is a cycle that continues to deteriorate. And I think it overly stresses. I'll be honest, I've always sort of wondered, since beech is really clonal, I've always sort of curious whether or not you get faster decline and faster mortality in saplings that are actually root grafted to a mother tree. Because if she detects stress, maybe she pulls back some of the, reserve from those underground root grafts. I don't know.
I wouldn't know that, but I think it's what happens when you have the fine twigs dying because the buds abort. Over time, you make the whole structure, the whole tissue you deprive the tissue of photosynthate, right? And so there's fewer defenses for those twigs against secondary invaders, and those secondary invaders are the ones that actually eventually cause a dieback. That's my guess.
That was a really interesting speculation about the mother tree pulling back the support of the clones, but
Yeah, I have no facts for that.
no, but that's a really interesting hypothesis. So for those of us who have not yet established monitoring plots, are there questions that have arisen since the template has been drafted that you'd like us to start looking at? Basically amendments to the DEC monitoring sheet?
Well, I mean, in the very beginning, you were making thoughts and projecting other potential data elements to collect. I mean, we use for our monitoring project, we do sort of assess some bit of worm population, but so I know you had mentioned maybe the amenthus and its presence, you know, anything that otherwise compromises, you know, forest soil and litter, you know, stresses additional tree health out. There's a lot of interest in moisture. You know, these are, you know, microscopic foliar nematodes, and they really need moist environments in order to move around and to re-inoculate the buds. And so I don't think anybody's really settled on exactly how to best capture, you know, moisture gradients or proximity to riparian areas. Enrico, you want to chime in?
Yeah, I can chime in a little bit because we've done some risk mapping based on environmental and climatic variables. And if you want me, I can share some data. It's a paper that's in progress, so it's not in the best of shapes, but I can show you some stuff. So this paper is being written at the moment. Let me just find it here. And I'll show you some data. With some modeling. Can you guys see this screen here?
Mhm.
So this screen just shows you the variables that we used. So we use aspect, slope, a digital elevation model, solar radiation, precipitation, relative humidity, blah, blah, blah. And after it all is all chewed up into a predictive model, we came up with a couple of risk maps. This is only for Northeast Ohio. Oops, what happened there? Sorry. Right here. This is only for Northeast Ohio. So it provides sort of an idea of the areas that are likely more prone to infestation by beech leaf disease going forward. It turns out that the best predictive value, the best predictive variable was minimum temperature. Okay, and then aspect and land cover classification and beech bark disease, beech, you know, excuse me, basal beech area and other things sort of came after. It seems like the minimum temperature is really the most important predictor. Having said that, we still don't know in which direction that goes. In other words, I still don't know if it is a lower minimum temperature or a higher minimum temperature that predicts distribution of the disease the best. We're still working through the analysis, so I don't have an answer to that. But it seems like minimum temperature is the critical factor here for some reason. I don't know. So that's all I can say at this point. We're doing more work in modeling, but I don't have any more data to share with you guys.
So how are land managers in Ohio starting to cope with this? I mean, are they looking at stockpiling money for takedowns in the near future, or what kind of steps have they taken?
Yeah, management is still a big giant question mark. We did start a couple of pilot experimental treatment projects. Yeah, I can share that too. In 2016, we started a project with Davy Tree using a polyphosphate 30 compound. It was one of our annual plots, and you can see this small tree here. This is all the bigger that the tree was. We picked fairly small trees, fairly isolated, had some BLD leaf symptoms, but not too severe. And you know, they came to me wanting to be able to trial this particular product, and otherwise there was nothing. And so we did this treatment. We did 2 soil injections each year, every year. And then, you know, severed any of the root grafts, etc. There were 40 trees total in the project, 20 control and 20 treated. And this sort of, again, is a bit on the timing. We did canopy assessments at the same time every year. So we do those canopy assessments in September. So as somebody already mentioned, sometimes heavy affected BLD trees start to lose their leaves in the midsummer. So in order to avoid that complication, we always keep our assessments to usually that first week of September. So the product is 2 ounces per inch. And this is just to kind of show you the size of our particular trees. In 2016, this very first year, this was part of our annual plot. So I have it disconnected here because some of the trees got included in the project and then some did not. And then we added other trees. So because they wanted, you know, 20 treated trees, we went outside of our only narrow plot to be able to find sufficient sample size for them. But as you can see, they're small trees in size. And basically what we have found, you know, treating these 5 years in a row now, twice a year, is the total canopy cover is greater in these treated trees than the untreated controls. So speaking to the, like Enrico said, the bud dieback, the twig dieback, that's kind of a continual process. And so even after the very first year of application, we see a significant separation between the canopy cover of the treated trees and the untreated trees. So that was a very promising project that we pilot started. They were all small trees, but we did expand that project to now using a soil drench application of the same product on large trees. So that only just started last year. In 2021 is when we treated those large trees to see if we would get a same kind of effect. Let me see. Oh, I go through a whole lot more data of this particular project. So I don't know if you want to see every nuance of the data for this particular application, but the bottom line is there's only a few projects, only a few treatments. We also partner with Bartlett on an experimental research project as well, using emamectin benzoate products, and then abamectin as well with Davy. But this, I'll stop here for a second because this kind of illustrates the cover, even in the mild symptom BLD banded trees, and then those heavy crinkled leathery leaves. The proportion of the canopy in each of those leaf symptoms don't really change through time, and they don't really change whether or not you're a control tree or a treated tree. So roughly 25% of the canopy of the available, you know, leaf canopy that's present will have symptoms, you know, and that's just always. And I think that speaks to the variability in that nematode population on a year-to-year basis. You're just, depending on what's re-inoculated, you're going to get some banding pattern every year, regardless of treatment. And again, because it's such a thinning process, you don't necessarily have dead main branches, but you have this continual thinning of the dead fine branches. So in the control trees, you see just this higher and higher proportion of dead fine branches every single year. So that continual stress. But dead main, and I showed this picture earlier, this is what that looks like. This is just because it's just that no a single dead main branch occurs. You just always have these random sporadic leaves hanging on for dear life at the end of every branch tip.
What kind of effects are you seeing in the understory? So one of the concerns we have is just with the gaps in the canopy, increased invasive species and ruderal species. We're concerned about water temperature increases in our vernal pools. We're wondering if, if you're seeing a jump or a kick to natural regeneration with other species beside beech, for example.
Yeah, that's an excellent question. I mean, I don't take soil temperature, although that's a really great idea to have looked at that changes because of that increased light environment. We do take canopy gap assessments, but we really haven't seen a huge influx or change in the herbaceous layer just yet. But to your argument, that was the same thing for us with ash trees. Like we have pumpkin ash. And so in our old oxbows or near vernal pools, we chose to treat those trees because we didn't want that canopy dieback and that increased light and that change in soil temperature and moisture and et cetera. So that was an area we chose to, proactively treat those trees. So that's why this project was also started, was trying to decide whether or not there were certain specimen trees that we wanted to try to keep, or in certain habitats where we felt like maintaining some canopy cover was important.
I must say that this phosphite treatment is one of the treatments that has most impressed me for trees recently.
Yeah.
For any disease, really. Yeah, I don't think demamectin is giving as much, right? Promising results. Is that correct?
That's true. And actually, I think that's my next slide. So this one that I'm showing you actually is a branch on a treated tree on the left and an untreated control on the right. So this is sort of that last illustration. This is why you have these branches that come down and then there's one leaf cluster at the very tip, right? So there was one bud that produced those 3 leaves and et cetera. So you have this really thinning kind of control situation. Let me fast forward. So close your eyes. Also with Davy. So Davy also decided to expand into the amamectin benzoate and abamectin trunk injections. So we picked an additional beech stand where we could try to do some of those trunk injections also just starting last year. So it's too early for results from that particular project, but they too wanted to be able to try a different treatment method. Bartlett tree, we did a project also with amamectin benzoate. In 2019, we started with some small trees, and by small trees in size and small trees in treatment application. There was only 5 treated and 5 control, and we got kind of mixed results.
Connie, can I just interrupt you for a second? What was
go right ahead.
What's the purpose of amamectin? What's it going after?
It's an insecticide.
Okay.
And I think that it's thought to also have some nematicidal properties. So any of the products that they're looking at, the abamectin, the amamectin, those are all going to be insecticides. I don't think anybody has or maybe Bartlett does. I was thinking of a fungicide treatment. Bartlett, since 2019, Bartlett tree has a couple of different projects scattered around. They have, we have some with them. Holden Forest and Gardens have some with them. I think they were doing some other treatments in Virginia. They're very interested in trying to experiment with a variety of products, a variety of concentrations, application rates. The translocation of the product is the big question, because if we think we have to target the nematodes or the nematodes inside buds, inside leaves, you really have to make sure that the molecular composition of the product that you're applying can get there. And so that's some of the different projects that I think that Bartlett's really working on.
Thinking out loud here, I'm thinking if, and that's a big if, but if the bacteria that we found are really evolved, trying antibiotics would be interesting if somebody was interested in trying that.
What was the, you might have mentioned it, what was the timing of this treatment? What season? What, that could probably factor in, right?
Yes, for sure, because you might be hitting different stages of the life cycle. So the Bartlett project that I think I have a graph for next, this was started in 2019. So we treated it in the fall with hopes that we would be affecting any nematode load in the buds for the following year. But that's another piece of the puzzle between Davy and Bartlett, that they're trying to figure out, when is the best time to do the application to target the nematodes at various stages of their life cycle. Okay, so again, very small sample size, but this was part of what it was. It's Andrew Lloyd was our PI with Bartlett that was working on this project with us. And so you can see it was mid-September of 2020 that we did this small project. These are the non-treated. We did a proportion of the canopy of asymptomatic striped, and then those solid or crinkled leaves. And so we tried to stay as pure as possible. There was an original design to this project that unfortunately, because of COVID, interrupted what we were going to get done. So now it's a little bit more like we treated the trees, now we're just going to have to monitor. It was a little more hands-off approach. But the very next year, the non-treated trees have a little bit higher proportion of striped leaves and solid leaves compared to the single application of the triage from the year before. Now, again, it's only 5 trees and it's a pretty small sample size, but that was their initial project. And they did then also try to take and look at nematode on other trials on some of their larger trees when they were doing applications of mectonite at 10 mils per inch, 20 mils per inch, and then they're untreated. And you can see it's highly variable. So there's still a lot of trial and error that they're navigating with their samples and their treatments.
Is Cleveland Metroparks considering fortifos treatments of selective trees in the landscape? So like near vernal pools, for example, or is it still just trial basis?
So when the polyphosphate treatment that we did on the small trees was so promising that we did choose large trees to treat. And the stand that we selected is actually one of our more mature beech maple forests. We call it our A.B. Williams Woods. And it's our flagship beech maple stand. So it is our priority location and was worthwhile. So it's still a fairly small sample size, but we did target the expansion of that project on species that and a location that was a high priority for us.
Is there any sign that there's transport of the fortifos or the polyphosphate from a mother tree to the saplings?
That's a good question. I would venture a guess and say no. The way they do the soil drench is really, I mean, they clear all the duff layer away from the trunk and they do a direct pour right around the base. And I would assume pretty much the fine roots get taken up by that mother tree and translocated up almost directly. I'm not sure, if it could, or how much you would have to use to then be grafted out to any of the clones. Guessing there though. So I think I'm stopping again. Any other questions?
Have you given up on trying to control spread? Boot brush stations, banning municipal leaf collection and compost or anything like that, or is it just too late?
Yeah, that's a good question. When we very first found it, and the very first stand that we came across it wasn't on our property. It was at Holden Forest and Gardens. We were helping them do another project. And I had the entire crew go home and wash everything, their ball caps, their backpacks, everything, because we had no idea. We use horse hoof picks to clean the boot tread, and we use either a 2% bleach or a Lysol to spray off boots and that kind of stuff. But if you think about the nematode, it would be really hard to spread to a new location and then have that nematode get to the bud if, you know, if that were the case. And the disease transmission, I'm not sure is as concerned the more we now know about that nematode. Other management recommendations, I know, and I don't know if Maria, you can speak to it at all, but I know that there was some experimental silviculture work that New York did to try to do some proactive cutting and thinning to sort of isolate a stand to see if that could help reduce some of the spread. But I think that was just done last year.
Yes, that was a very, very small 2-year project that we did. But it really wasn't big enough to make any sort of conclusions from, especially on spread. That wasn't really like the method of spread wasn't something that we looked into with that. But it's something that we're also interested in figuring out if possible.
I can maybe add a couple of things to this. You know, hot off the presses, speaking like between yesterday and today, essentially, we've done a microbiome analysis of roots at some of the trees that Connie has been monitoring. And we did not find any evidence, I'm talking about molecular evidence of nematodes, little encas or otherwise, in the roots so far. So that's one thing, which is at least one piece of good news. I think it's unlikely that the nematodes will move around in people's boots and things like that, but you never know. I mean, precautionary measures are always good. The other thing is that we don't really know how this nematode moves around, as we've said. There is a possibility, though, that so first of all, it's been found in association also with mites. And mites may act as vectors because the nematodes by themselves, they can't move very far, right? They wiggle around, and so there's mites, and then there's a possibility that the birds may be vectors either by way of mites or directly, especially if they eat the buds, and some birds eat beech buds with a vengeance, actually. So there's been some work done on beech bud feeding by birds. So anyway, we don't know, but there are various options for these nematodes to move around. And at this point, it really seems like this is an exotic nematode in North America. So how does it spread so far and wide? Again, we don't know, but birds might be a good way to go around. It seems like if the map that Connie has shown you actually represents spread, and we don't know that. We don't know if it's actual spread or if it's just people looking in new areas. But if it is spread, it seems to be sort of in a west to east direction, because it was first found in Ohio. And the nematode tends to be located in areas near big bodies of water. That's another thing that we sort of have noticed. So either the ocean or the Great Lakes. But on the other hand, there's distribution across Pennsylvania, right? I mean, from Ohio through Pennsylvania and into New York and so on. So there's so many unknowns. I don't even know where to start sometimes, but these are some of the thoughts, some of the speculation, some of the evidence we have. And this is all we can say, really. I don't know, Connie, if you have anything to add to that.
No, there's lots of questions. We wish we could give you better answers or more answers, or, happy to be helpful or resources. You guys develop your working group or come up with questions. They're questions that have been in our brain now for several years. So for what it's worth.
I would love to have tons and tons of money to do tons and tons of research. So if your organization can do fundraising, let me know. I'll help.
I have one quick question. You mentioned timing of assessments, and that's something that we've discussed as well in our group. I was just wondering how significant you think that is when comparing assessments across the region. If like you can confirm that Cleveland Metroparks is doing it at the same time, but you can't confirm, that maybe New York or where else is doing it at the same time. Just wondering what your thoughts are.
So two thoughts. One, thanks for the question, and then two thoughts. One is we know that the beech leaf disease symptoms are immediately visible right after bud break. So you can start doing an assessment as soon as they're in full leaf out. And that condition really doesn't change throughout the growing season. So if a leaf leaves out with just 2-3 bands in May, it's going to have 2-3 bands in September. That doesn't necessarily change. But two, what does change is in heavy affected trees, those advanced leaf symptoms, the really dark crinkled leaves, those can fall off. So starting in around June, July, those leaves can fall off. And then it depends on what proportion of the canopy has that kind of leaf, which is why, we started our assessments. And it was by convenience of crew. They were working on other projects initially. So we did all of our beech leaf disease assessments end of August, first week or two of September. And that's just how we stayed consistent. And then we started finding out through another paper that we published was the fact that we have early leaf drop in those heavy advanced leaves. So that's how, you just need to make sure that whatever you choose, you do at the same time, because in heavy expression trees, you could lose part of the canopy due to early leaf drop.
What's your methodology on canopy cover? What are you using to measure your canopy cover?
So it's the same in the monitoring protocol that Maria has as well. So it's a rank cover value for total canopy cover, which is why if you saw on my scale, so much of a full canopy tree is somewhere around 75 to 87%, because we take the midpoint. Anytime I do the graphs, it's the midpoint of that cover value range. So 75 to 95, and then it's like 95 to 100. And then I use that same rank value cover range for any of the leaf symptoms. And I'm happy to share that. It's very similar to what you'll find in the Tree Health Survey app. It gives you a drop-down list of what proportion of the canopy is in what cover range. So they're broad, it's a doubling factor. 10 to 25, 25 to 50, 50 to 75, that kind of, those large range.
Yeah, I didn't know if you were using densitometers or anything like that to measure, to get your, averages.
So we will use a densitometer for light gap. That is our light gap measure. Not everybody does though. So it's in our monitoring protocol, but I know that state to state, they did not all incorporate densitometers in their plot surveys, but we do.
All right. So out of respect for everybody's time, we're going to wrap up, but thank you so much to Dr. Bonello and Dr. Hausman. That was incredibly informative, kind of depressing as we expected, but really good to hear your experiences and that 10% mortality of young trees number that you showed gives us something to work with, I think. And if you have any suggestions for us, going forward, things we should look for, things we should consider when establishing our plots, we're super happy to help and hear anything that you have to offer.
Well, thanks for having us. I dropped my email in the chat if anybody wants to. You're welcome. We'll share whatever we got. Hopefully some of it'll be better news in the future.
Thank you very much, guys. Again, appreciate it. Taro, do you anything you want to say to the group before we?
We'll try to meet again in 2 weeks.
Okay.
All right. Thank you again.
Thanks.
Thanks.
Bye-bye.